Hello everyone. It's on time, and then the chairperson, Dr. Homburg, will be somehow late, so I want to start on time. So thank you for joining this webinar, and I'm glad to be here to talk about the clinical phases in neurorehabilitation. So in neurorehab, the evidence becomes more important to make a guideline and get a new indication, and the most importantly, for patient better recovery. So in today's talk, I will introduce some basic concepts for clinical trial phases based on drug trial, but also I will talk about the trial with behavioral intervention or medical devices trials. I try to also present some relevant examples for each trial phase. I hope my talk will be very helpful for the new researchers. And then I want to start now. So Christian, can you please start my video now?
I'm glad to be here for this webinar, and my topic today is clinical research phases. I have no conflicts of interest related with drugs or medical devices introduced in this talk. Today, I will introduce some basic concepts in clinical trial phases. As you know, the clinical trial phases were from pharmaceutical studies, but this concept can also be applied to behavioral intervention or medical devices. After introducing the basic concept, I want to present some specific examples in the neurorehabilitation research. This is the drug development timeline. There is a number of compounds for clinical use, but those compounds have to be tested by sequential clinical trials to prove their efficacy and safety. The success rate to reach commercialization is usually low. The clinical trial phases are usually composed of 4 phases, although phase 0 is sometimes included. The preclinical drug trial usually means in vitro or in vivo animal studies. In this preclinical trial, we can get the pharmacokinetic and the pharmacodynamic and then toxicity data, and the safe starting dose for trials in humans can be estimated from clinical trials. Phase 1 is usually a short-term, the first human trial with a small sample size, usually tens of participants. The goal is to generate pharmacokinetic and the pharmacodynamic data in humans and to determine the maximally tolerated dose. In this phase, usually healthy volunteers participate, but individuals with target diseases can be included according to the situations. The usual study design is a single group, observational, or free post-study design with a single ascending or multiple ascending doses. The goal of phase 2 trials is to confirm safety and to explore efficacy. Therefore, more participants are needed, and patients with a specific target disease will be recruited. Recently, a small randomized controlled trial is usually conducted as a phase 2 clinical trial. A phase 3 clinical trial is usually required for FDA approval, but phase 2 trials can sometimes become pivotal trials. Phase 3 is the pivotal and large-scale randomized controlled trial with broader eligibility criteria than a phase 2 trial. The major goal is to confirm efficacy and safety. It requires a longer trial time, usually several years. Phase 4 is the trial for FDA-approved indications, and the major goal is to evaluate the long-term safety and effectiveness with a more diverse target population. Most market surveillance is one of the forms of phase 4 trial. So in summary, page 1 for human pharmacology, page 2 is for exploring therapeutic efficacy, and page 3 is for confirming efficacy. During drug development process for FDA approval, pharmaceutical companies have to submit an investigational new drug. And after the successful phase 3 trial, they submit a new drug application, that is NDA, for FDA approval. This process, or the term used may be different between countries. Phase 2 is sometimes further divided to phase 2A or phase 2B. Phase 2A usually is called proof of concept, preliminary, or pilot studies, and the traditional goal is to assess dosing requirements. Researchers can use the surrogate endpoint rather than the clinical endpoint. For example, in the motor recovery trial with a new drug, fMRI findings or transcranial magnetic stimulation results can be used as a surrogate marker for motor recovery. And phase 2B is more specifically designed to study efficacy before phase 3 trial, so usually with a larger sample size than phase 2A trial. Phase 3 can be further divided to phase 3B, and this means clinical trials after regulatory submission of new drug application, but before approval. This slide shows the drug success rate from phase 1 to FDA approval according to disease category. As you can see, the drug success rate in neurology is low, and it becomes even lower in the neurorehabilitation field. From the next slide, I will show some examples of drug trials in the neurorehabilitation field, and most of the trials used the approved drugs for other indications, which is off-label use. Some of the trials are led by pharmaceutical companies for new drug application, but as you know, most of the drug trials in the neurorehabilitation field were investigator-initiated trials and were phase 2A or phase 2B trials. The first example is botulinum toxin type A use for spasticity. The Botox was first approved for blepharospasm or strabismus in 1989, and the company has expanded therapeutic indications with the clinical trial, and as I know, the FDA approved the botulinum toxin type A use for spasticity around 2010. And then for the adult lower limb spasticity, FDA approved the Botox around 2016. Preliminary studies for reducing spasticity were conducted, and the Botox for upper limb adult spasticity was approved by FDA based on these 5, maybe phase 2 clinical trials. For FDA approval, usually large-scale multi-center phase 3 trial is required, but in this case, Botox was widely and safely had been used in other diseases, and the action mechanisms of Botox to reduce spasticity were clear. So these 5 phase 2 clinical trial results can support the FDA approval. Regarding adults lowering spasticity, several investigator-initiated randomized controlled trials, which may be phase 2A or 2B trial, had been conducted and the therapeutic efficacy was suggested. After these phase 2 clinical trials, the phase 3 large multicenter sponsor-initiated trial was conducted to confirm the therapeutic efficacy and safety. The positive results of this phase 3 study led to Botox indication expansion to low-limb adult spasticity. This clinical trial is also for the adult upper limb spasticity, but this trial was started after the FDA approval for upper limb spasticity. Therefore, it was not a trial for new drug application. Therefore, the Botox use when this trial conducted was on-label use. But the authors want to know that if Botox is more effective when intensive arm rehabilitation is provided together after the Botox injection. As you can see in this cursor diagram for this botulinum toxin study, this was a multicenter randomized controlled trial with a moderate sample size, which may be usually considered as a phase 2 trial. The total subject number is 140. But in this paper, the author stated that this was a phase 3 clinical trial because the same authors conducted a similar small sample-sized pilot study before. In this case, the first study can also be categorized to phase 2A and the second study to phase 2B maybe. If this was a sponsor-initiated trial, this trial is conducted after the approval by FDA using the Botox for spasticity. It can be categorized to a phase 4 clinical trial. So the exact phase categorization of clinical trials, especially for investigator-initiated trials, can be difficult and sometimes is not clear. The second example is a selective serotonin reuptake inhibitor and stroke recovery. As you know, SSRI was already approved by FDA for depression before these following clinical trials. And in previous animal studies, which may be a preclinical study for SSRI used for stroke recovery, many kinds of antidepressant, including SSRI, showed positive results in stroke recovery. Based on data from clinical studies, many randomized controlled clinical trials using SSRI for stroke recovery were conducted. As you can see here, some trials have a very big size of sample, but usually around 100, or very small sample size around 50 or 40. So most of these clinical trials can be categorized to phase 2A or 2B trials. And then, as you can see here, from the meta-analysis with the phase 2 studies, it shows some beneficial effect. Everybody may know this clinical trial. So among these phase 2 clinical trials, the landmark well-designed randomized controlled trial using SSRI for stroke recovery was FLAME trial, which can be categorized to phase 2B trial. The total sample size was 118, and they showed better motor recovery in fluoxetine group, even after adjusting the presence of depression. And then the phase 2 clinical trial for FORGE is to approve the safety again. And as you can see in this table, The adverse events were also comparable between Prozatin and placebo groups. So this FRAME trial showed the exploratory efficacy. And this sample size clinical trial cannot confirm the clinical efficacy. So we need a phase 3 trial based on the preclinical studies and phase 2A and 2B trials, and also based on meta-analysis. After the FRAME trial, and then phase 3 clinical trial was conducted. As you can see here, similar clinical trials were conducted in different groups. They used fluoxetine for functional recovery after acute stroke. So this is a multi-center and very large sample size clinical trial, which may be a definitely phase 3 clinical trial. So in this talk, I will not discuss about the study result. But anyway, they showed a negative result for SSRI use for motor recovery. This study is also very famous. The amantadine use in traumatic brain injury is also similar cases as SSRI trials for motor recovery. As you know, the amantadine indications are treating influenza A virus and Parkinsonism. So amantadine use for TBI consciousness recovery was off-label. As in the study of SSRI, the active researchers had conducted several phases 1 and phase 2A clinical trials and suggested the possible clinical efficacy of amantadine. After that, these larger-scale multi-center RCTs showed the clinical efficacy of amantadine in severe traumatic brain injury, especially for recovery from disorders of consciousness. We can categorize this study to phase 2B maybe, I think. But if you think the results of this study is confirmatory for amantadine efficacy with enough sample size, it can also be categorized to phase 3. Again, the categorization of investigator-initiated trial according to the concept of clinical trial phases is sometimes not clear. Then let's think more about the traditional rehabilitation interventions or behavioral interventions. This is the table from the article written by Hardy et al., which was published in PMNR at 2012. I think it's worth reading. And in this table, you can see the not the between group study design. This is the within group study design. And then this also can be the research design, even though it's not the strictly clinical trial. Or if we want to derive the rationale for post-clinical researches, we need the kinds of clinical trial. For example, pre and post test study design. Usually, we recruit the patients with chronic stroke, and then we think they are very stable for the stroke recovery, and then we can sometimes apply this kind of study design, even though it has a limitation for internal validity of the study. And then we can also use historical control, usually for the quality improvement activity in the hospital. And then futility study, which is comparing the treatment effects with preset criteria. And then sometimes we can use observational studies, which may be case control, cohort, or registry-based studies. So we cannot directly apply the concept of pharmaceutical clinical trial phases to the rehabilitation trials with behavioral interventions. But I think the conceptual studies can be corresponded to phase 1. Some examples are observation studies like this, including the cross-sectional case control studies in patient group, not the healthy people. And to prove the preliminary efficacy, we sometimes use the simplified study design rather than randomized controlled trials. This can include the pre-post single-arm design or using historical control or this kind of futility study with comparing treatment effect with preset criteria. Even though this study has problems in the internal validity due to various reasons, I think these studies can correspond to page 2A, where it can be located somewhere between page 1 and page 2A. Recently, the trials using behavioral intervention in neuro rehabilitation field usually use the randomized control study design. So this RCT can be categorized to page 2 or page 3 clinical trials according to their sample size or statistical power and the study purpose. Sometimes crossover study design can be considered as an exploratory pilot trial in the neurorehab research. And this can be categorized to phase 2A study maybe. And usually for the crossover study design, the patients with stable state, such as chronic stroke patients, are usually recruited for this kind of crossover design. So when you think of the trials with behavioral intervention in neurorehabilitation, the subjects are usually patients with specific diseases such as stroke, traumatic brain injury, or Parkinson's disease. The behavioral intervention are usually a low-risk intervention, so classical phase 1 trial with healthy people can be usually skipped. But if the intervention is new and researchers do not know the effect size or their presumed clinical efficacy, the researchers can plan a study with healthy people to investigate their preliminary efficacy. For example, in this study by Gamari et al., they developed their virtual prism adaptation therapy using immersive HMD and applied the randomized control trial design in healthy subjects like this. So total 48 healthy subjects were randomly allocated to no deviation group and the 10 to 30 degree prismatic deviation group. So in this case, this kind of trial may be categorized to phase 1 trial, even though they use the randomized control trial design. And in this study, the authors also use virtual prism adaptation therapy, but the target population was a stroke patient with specific impairment, which is unilateral spatial neglect. So this target population is very specific, disease specific, and impairment specific. So it's a phase 2 trial, maybe. And then they use 3 conditions, and then the design was not a parallel design, and this was the crossover design with a washout period more than 2 days. So I think it's a phase 2A clinical trials to prove the preliminary efficacy of virtual prismatic adaptation therapy, which was a new intervention. The last example for behavioral intervention is AVERT trial, which is a very early mobilization trial after acute stroke, which was very famous. To derive the further clinical trial rationale, which is a prospective interventional trial, this group may first conduct the observational studies and reported that many stroke patients were inactive, and the activity level is not uniform between hospitals during acute stroke care. I think this kind of observational study may correspond to the phase 1 trial. After these observational studies, this group conducted phase 2 randomized controlled trials with 71 patients with stroke. And then they implemented a very early mobilization therapy and they compared efficacy and safety with the standard care group. And then they showed the feasibility of very early mobilization in terms of earlier timing of mobilization and then more total dose of mobilization in their very early mobilization group. And then they also showed preliminary efficacy, which shows a better MRS outcome in their very early mobilization group after adjusting for some confounders. Based on a phase 2 clinical trial, this group designed the phase 3 trial, and then everybody will know this study result very well. This is very large sample size. For the behavioral intervention, I think it's the largest trial in the neurorehabilitation field. This slide shows the success rate of drug trials in each clinical trial phase. The success rate in phase 2 is the lowest. The trial in the neurorehabilitation field, we can find many phase 2 trials with positive result, but the number of phase 3 clinical trial is small. So based on the positive result in phase 2 clinical trial, and then positive effect in the meta-analysis, recently the large-scale multi-center trial for the specific intervention was conducted, and the results have been reported. But unfortunately, many of them showed negative results. So I don't know why. What is the truth? But there are some reasons why it is very difficult to get positive result in phase 3 clinical trial in the neurorehabilitation field, especially using the some kind of neurobehavioral intervention trial. Lastly, I will briefly discuss about the medical device clinical trials. FDA classifies the medical device to 3 classes according to their risk. This classification may be different between countries, but may be similar between countries. Not all medical devices require clinical trials. Usually, the class 3 medical devices with high risk usually need clinical trials for a pre-market application, which is PMA. And then the class 3 medical devices include pacemakers, stents, heart valves, or orthopedic implants. You can see in the class 1 is very non-invasive, which may be adhesive bandages or tongue depressors. And class 2 is between class 3 and class 1 risk. And then for the risky medical devices, which may be class 3, or sometimes maybe class 2, usually class 3 medical devices, IDE, which is an investigational device exemption, is required for clinical trial. So FDA approval of an IDE is required for United States human study of a significant risk device, which is not approved for the indication being studied. So every researchers should know this kind of medical device classification, and then they have to design their study according to this medical device classification. And sometimes, even though it is IIT, investigator-initiated trial, they need IDE, so it was very burdensome for clinical researchers. So anyway, if they violate the regulations, is a big problem. So very well understanding is necessary. In medical devices trials, a preclinical study can be usually skipped, but the following studies can correspond to preclinical study in pharmaceutical or clinical trials, which may be a laboratory testing to the performance or safety, usually using the prototype, and then engineering and then quality testing or computer simulations. And the animal studies are sometimes required, but are not mandatory in many cases. We can categorize the medical device trials like drug trial phases. And first stage is early feasibility study with 10 to 30 subjects. And the purpose is to get human data for preliminary safety and device performance data. And this study can guide device improvement and the future study design. And the second stage is traditional feasibility study with maybe 20 to 30 subjects using near final or final device to explore safety and efficacy. And then the pivotal trial is with a more sample size, and then it's proposed to confirm clinical efficacy and safety, and in result to get the FDA approval. Compared to pharmaceutical trials, the device trials tend to be a small sample size and are difficult to blinding or sham controlled, and have a higher rate of technology change during trial. And it depends more on precision technique, and it can have more diverse endpoints. There's a difference between the pharmaceutical trials. This is one of the example of pivotal clinical trial for PMA in Korea for using the rTMS for motor recovery after subacute stroke. The sample size was 77, which was less than drug trial for IDA. And then this rTMS study, sham mode can be applied, but the trial with medical devices requiring invasive procedures, usually SANE control and blinding is very difficult. In this webinar, I just talk about the basic things to know about clinical trial phases, and then I presented some examples of clinical trials with the pharmaceutical trials, and then trials with behavioral intervention or medical devices trials. Anyway, the classical clinical trial phases are based on pharmaceutical trials for FDA approval, but the trials with behavioral intervention or medical devices also have similar trial phases. According to the drugs or intervention, the trial can start from each different phases. Therefore, in the neurorehabilitation research, it is recommended to follow some kind of following trial development framework. So based on the pre-existing evidence, and then based on novelty of intervention or funding, any kind of feasibility, and the clinical pages and study design has to be decided. So I believe that the audience can learn more valuable knowledges from the upcoming webinars, started with the study designs, which is very important. Thank you for your attention.
Okay. So thank you, Professor Kim. That was an excellent lecture. I must apologize. I was still in the emergency room, and therefore I couldn't join you right from the beginning on. Now let me have a look at the chat room, Christian. There, at the moment, I see only one question from Dr. Guryaev, and stating that it's a little bit about parametric versus non-parametric statistics. And of course, that is critically dependent on, well, the sort of parameters you use, and the sort of scalings you use. And of course, with all these trials, we always have to be pretty careful to apply the proper statistics. There is an old joke, say statistics, bad, they say lies, bad lies, statistics. And with statistics, of course, you can do, well, you can push data in various directions. And proper statistics are, of course, absolutely mandatory to have to tell really the truth from what data are showing. So there is no, I think, no categorical answer to that. It really depends on the design, and really it also depends on the sort of scales used. Professor Kim, would you like to comment a little bit on the interaction of parametric versus non-parametric statistic approaches?
Oh, it's not the scope of our lecture today, but anyway, the dependent variable, so outcome data, so the characteristics of outcome is maybe important. So if it is categorical, we have to use some kind of light statistics, and if it is continuous variable, we have to check it's have a normal distribution or a non-normal distribution. According to that kind of normal distribution, we can decide whether parametric statistics or non-parametric statistics can be used. So there, Dr. Guryaev question may be usually in the neural rehab trial, most of the commonly used outcome is a modified ranking scale or distributed rating scale, which may be categorical variables. So, but in some articles we may review that in some articles they use MRS like a continuous variable, and sometimes suggest some average and standard deviation. So I think it's somewhat problem, but it's rather than use the make the binary MRS to the binary variables, that is a favorable or non-favorable outcome, then we have to get their two-level dependent outcomes, and then you can apply the right statistics. So how about your opinion about that? So Dr. Homburg?
Yeah, well, of course, it's always wise to have a good biostatistician consulting the researchers on what you're doing. By and large, non-parametric statistics are usually much more conservative than parametric statistics are. And of course, one has to be very careful in using parametric statistics in data ensembles in which you do not know about the actual distribution. And especially when it comes to categorical data as MRS, as FIM, and so forth, non-parametric statistics may be better and more conservative. Let me nevertheless allude to another point, I think, which is very important. And when it comes to clinical trials in neuro rehab in general, you know, all the, let's say, theoretical framework of evidence-based medicine was originally derived from primarily pharmaceutical studies. And the characteristic of these pharmaceutical studies is usually that it's a simple question. If you use 50 milligrams or 100 milligrams of aspirin for secondary prevention of stroke, that's a pretty straightforward answer. And you just sort out how many strokes are occurring over a particular period in time in one or the other group. Pretty simple. Nevertheless, you need thousands of patients involved to come to a conclusion. And the problem with, let's say, clinical trials in neurorehabilitation is usually that you have much smaller sample sizes. The sample sizes, well, the AVERGE trial, one was commenting on, was one of the biggest trial ever done in the history of neuro rehab with a couple of thousand patients. But most of the studies have much smaller sample sizes and maybe below 100 or several hundred the most, which of course makes it much more difficult. And the other point is that the heterogeneity of the databases is much higher. So that makes it sometimes very, very difficult to use the classical EBM designs and the classical trial designs in neuro rehab trials. And we have to keep that in mind, that eventually, depending on the question you want to answer, you have to go for other statistical, non-parametric, or single case designs, even crossover designs and things like that. That makes it much more complicated in neuro rehab to talk about statistics. Let me briefly look if we have more questions. There is a person, anonymous, whoever it may be. Could you please talk about what are outcome measures and how they developed, or at least a general view? Okay, outcome measures, of course, are extremely important. I think probably Dr. Kim is also willing to respond to that. And the second question is, what is the minimum data size, the sample size for pilot studies regarding swallowing testing, for instance? Okay, would you like to comment on this briefly?
Specifically for the video fluoroscopic study versus swallowing therapy, maybe, do you mean the...
Well, then I think the question is related, what is the minimum data sample size for sort of a pilot study when it comes to a question as swallowing using video fluoroscopic studies versus swallowing therapy in pilot study? Well, you know, when it comes to explanatory trials, I think usually you have to deal with smaller sample sizes, that's for sure, but for simple practical reasons. And well, depending on the contrasts in the study you expect, you have to find a way, what is the minimum sample size? There are mathematical models that. What is the minimum necessary sample size to come to a, let's say, result, considering the dimension or the size of the contrast between experimental and control group, for instance, you would expect. That is always critically dependent on the sort of measures you're using. And I think it's always a good idea, at least do some explanatory, to gain some explanatory data before you go for a real efficacy study, for instance. Just to have an idea how the size of the contrast between the experimental and control group will be. That is absolutely mandatory. And you may end up and say, well, the contrast is pretty low. And then, of course, you need much higher sample sizes. And you should add another 25% more than the minimum to be expected from a mathematical model to come to that. Another question, and I think which is very important, and you know, I'm running that for 3 decades. A very important problem in clinical trials in neuro rehab is inclusion and exclusion criteria. You know, every scientist tries to be very strict with inclusion criteria to make the study as scientifically neat and sound as possible. The problem is that if you do that, you may end up with the desperate situation that you simply do not find patients because these patients are not existing. And of course, it doesn't make sense to run a study on patients who are not existing at all. So we have to be very careful with inclusion and exclusion criteria. So also clinical trials in neuro rehab have to address the real world situation, because otherwise you do a good study, but nevertheless you do not answer a relevant clinical question, because the clinical question is related to really existing patients. So if you restrain yourself too much, too strict inclusion exclusion criteria, you may end up with a lot of recruitment problems. It may take years before you reach the envisaged sample size. And the question is that meaningful at the end? You know, so that is also a tough problem. That is also related, as what I mentioned earlier, to the problem: is this type of statistics we use in clinical trials usually? And this was more or less borrowed from bigger pharmaceutical trials originally. Is this the proper statistical method to use? And you know, there are alternatives to that, and also in this series coming up, we will address these issues in detail. So it's a tough problem. It's a tough problem, and using statistics in neuro rehab, by and large, is much more, let's say, problematic than using it in straightforward, let's say, pharmaceutical trials, where you have a very simple, clearly defined endpoint. We were lucky years ago when we were looking at sotorasib, for instance, in stroke, that we had a fairly good contrast coming out of this, which is nice. But usually, especially when you look at training techniques, not at pharmaceutical interventions, but at training techniques, the contrasts are much lower. For instance, when you look at the effect of, let's say, intensity of treatment on impairment recovery in stroke, you know, the contrasts are much lower. Let me have a look. Wangsuk, do you want to comment on this right away?
So I agree with your opinion, absolutely. So for the clinical trial, especially for the neurorehabilitation, the population is very heterogeneous, and then according to the study population, the intervention effect will be different. So targeting the appropriate population may be very important.
Yeah.
And also the behavioral interventions is a very complex intervention, so not the drugs. So the tight control is very important. So even though we think that the intervention is provided well, but if it is a multi-center trial, the intervention itself can be very heterogeneous. So I think it's a very difficult one to be addressed in neurorehabilitation trial.
Yeah, and I think in future, my gut feeling is, we have evidence that to some extent. My gut feeling is that we have to rethink a little bit the mathematical modeling of what we are doing. And I think that will be of increasing importance in the future in the field of neuro rehab trials. I'm absolutely convinced, and there's no doubt about that, that we need decent clinical trials to find out what is efficacious and what is not efficacious. On the other hand, we know that we are dealing with very heterogeneous populations. And for particular questions, it can be better to use single case designs with a ABAB switch between interventions, for instance, in order to clarify if a particular intervention is helpful in a particular patient. You know, Barbara Wilson, she is a really very eminent British neuropsychologist, written a lot of books on treating of cognitive disorders, especially memory and others. And Barbara, for years, always said, let every patient be a single case trial. Okay. And I think that is a very good idea. If you're dealing with a patient with a particular problem, and you do not know what sort of intervention to choose to treat this patient, what you can easily do is do a baseline, use intervention 1, do baseline again, use intervention 2, go back to intervention 1, and do it in everyday clinical practice, just to find out what sort of intervention may be helpful. And I think I really liked, all through my career, I must say, I like Barbara's idea on this. And, you know, that is not something you can publish easily, but it's extremely helpful in day-by-day work, right? Which is a different issue. Okay, What do you think? Let me see if there are more questions. Probably there is. Yeah, there's another question. Are multidisciplinary studies available in neurorehabilitation? Well, Dr. Kim is probably willing to answer that.
Multidisciplinary studies available. Yes, usually the neural rehab intervention is multidisciplinary. So every day our practice is very multidisciplinary. So if you mean the multi-center studies, I think yes, it depends on your situation. So study population and funding, some kind of other feasibility may be a factor if you can do the multi-center study or not. So anyway, I think multidisciplinary studies may be available in neural rehabilitation. Yes?
Well, I think neuro rehab is always multidisciplinary.
Yeah, yeah.
Right?
Yes.
And I think probably what the question means is multi-center studies. And of course, we need multi-center studies for the simple reason that we need bigger sample sizes and that we have to reduce by bigger sample sizes. We have to reduce the heterogeneity of the data. But coming back, it's a good example. You mentioned Julie Bernard's AVERT trials. And this was probably the biggest ever done trial in neuro rehab history. Unfortunately, the main result was a somewhat confusing one in the sense that pretty early 24-hour starting rehabilitation was worse than doing it a little bit later, waiting a little bit longer. That has given rise to an awful lot of confusion. And although this was an extremely well planned, an extremely well performed trial, nevertheless, the final result of the AVERT trial was a little bit more or less disappointing and confusing. And that simply shows the dilemma we are in, because it's unlike 50 or 100 milligrams of aspirin. It's a different problem, right? And we have different. So what Julie and her co-workers in the AVERT trial did was to look at early out-of-bed rehabilitation in patients within the first 24 hours or later. And that was just on truncal activities. This does, of course, not at all mean that early rehabilitation is wrong. And that is another question. What sort of conclusions you are allowed to draw from a particular trial, even if the trial, the AVERT trial, was planned very well. Nevertheless, you may end up with an awful lot of unsolved questions. Well, there was another question on neuropathy, leprosy neuropathy. I cannot answer that. I don't know if Professor Kim is going to answer about that. I don't understand the question completely about neuropathy and leprosy, and probably I was 10 minutes late, so I probably missed something. So would you comment on this?
So I don't know much, but how can you define the neural rehab? So usually I think neural rehab is for some central nervous system problem, but if you can extend that patient to peripheral nervous system, yes, maybe. So it's a difficult question.
Yeah. Okay. I think what we have to conclude definitely, and that is sort of the take-home message, is that applying using trials in neurorehabilitation, first of all, is extremely important to do. Second, it can be cumbersome to plan it in a proper way, avoiding too strict inclusion-exclusion criteria in order really to address trials to real-life populations, because that is what we need. We need information on the selection of therapeutic options and what we can do. And number three is that, of course, we have to think about alternative biometrical methods, which are a little bit beyond the classical framework of the pharmaceutical trials, which were more or less, let's say, the milestones of evidence-based medicine when Archie Cochrane started with this idea of evidence-based medicine. But today we have the grade system, which is a little bit beyond that. We have other statistical options, looking at real-world data, at single case analysis, and all of this, I want to emphasize, will add to our knowledge. And there is another aspect, I think, which is important. I think all of us are very much in fond of evidence-based medicine. I think there's no alternative to that. On the other hand, I think, and I always try to emphasize that with my students and my junior colleagues, for instance, there is a world beyond evidence-based medicine, and that is custom-tailored medicine. You know, and in evidence-based medicine, usually is based on group data. What we're dealing with as clinicians on a day-by-day basis are individual patients. And of course, I mentioned that we can get data from individual patients in ABAB or single case designs, but the art of neurorehabilitation is to integrate the knowledge primarily derived from, let's say, evidence-based things into a custom-tailored, individualized problem. And that is a difficult, pretty difficult issue. And there are no, let's say, there is no primary route to do that. And I think that has to be very carefully analyzed. And I very much appreciate that in this series of seminars, we will further address these issues because they are really relevant to everyday practice and our thinking. You know, running trials necessarily also influences our thinking about how we deal with neurorehabilitation. And I'm very grateful to Mansukh that he mentioned the problem with, let's say, assessment of technical systems, bringing technical systems into action. There are multiple aspects, the security aspects, feasibility aspects, and of course, efficacy aspects as well, right? And that is still a little bit different from running, let's say, an interventional study using a particular technique or using a particular drug, for instance. Okay, let me have a look at the chat room if there are more questions which we have eventually not answered yet. Christian, can you help me? Is there anything else? I can't see it. Probably not. So we had about 30 to 40 people with us this afternoon, probably more. It's always a little bit difficult with the Zoom technology to know how many people were online. You know, all of this is recorded and can, at least a couple of days later, be looked again at the WFNR website. Let me thank Professor Kim again for this very excellent talk. Mansukh, it was a pleasure to have you with us. And in about a month's time, we have the next lecture. Did you mention that already? You'd see it on the WFNR homepage. We are doing at the moment 2 different series of lectures. One is related for young clinicians on methodological aspects, and the other one is more a general one. And this is related to all sorts of let's say, problems in neurorehabilitation. Probably one of the next one will be related to low-tech interventions, another will be related to high-tech interventions, and another one will be related to language and speech problems. So thank you very, very much. Let me look finally at the chat room again, if there's anything. No, I can't find anything new. So thanks all of you again very much for your kind attendance. Stay with us, look at the website, and we look forward to see you again on one of the WFNR webinars. And many, many thanks to Mansukh Kim for this excellent lecture. Thank you so much. Bye-bye.
Bye.